Nervous tissue is discussed in this screencast, which may be found in Chapter 7 of your textbook. This screencast was designed to help you achieve the following objectives. Describe the structure and function of glial cells of the central nervous system. This includes the astrocytes, microglial cells, ependymal cells, and oligodendrocytes. The second objective is to describe the structure and function of glial cells of the peripheral nervous system. These cells include Schwann cells as well as satellite cells. When we looked at a slide of nervous tissue under the microscope in lab, we observed in our visual field something that looked like this. There are two main types of cells in nervous tissue. The large star-shaped cells are called neurons. Neurons generate nerve impulses, but they lack the ability to replace themselves if they're destroyed. For the most part, if a neuron is destroyed, it is gone forever. Notice all the little nuclei that surround the neurons. These are nuclei of a second type of cell called neuroglial or glial cells. Glial cells outnumber neurons somewhere in the order of 10 to 1. Neuroglial cells do not generate nerve impulses. However, they do have the ability to replace themselves. The function of neuroglial cells include protection, nurturing, and support of the precious neurons. We will now look at each neuroglial cell, its basic structure, function, and will also identify in which part of the nervous system it is found. There are 4 neuroglial cells found in the central nervous system. The most abundant, shown here in yellow, are the astrocytes. The astrocytes are star-shaped. Astro means star. They have these little extensions that attach to nearby neurons and also to nearby capillaries. And they hold the capillaries, which provide the neurons with oxygen and with the nutrients they need, in close proximity to one another. Astrocytes also form a barrier between the capillaries and the neurons, which is called the blood-brain barrier, which we'll talk about later on. Astrocytes also have the ability to control the composition of the extracellular fluid around the neurons because they have the ability to absorb and degrade neurotransmitters as well as to absorb excess ions that would otherwise interfere with the normal activity of neurons. Another neuroglial cell of the central nervous system is the spider-shaped microglial cell. The microglial cell is a phagocyte. Since your immune system does not have access to the central nervous system, the microglial cell is responsible for disposing of any bacteria or microorganisms that invade the central nervous system. They also are responsible for disposing of any remnants of dead body cells. Can you think of why white blood cells of your immune system are not allowed access to the central nervous system? A third neuroglial cell of the central nervous system is the ependymal cell. The ependymal cells line the various cavities of the brain and spinal cord. They are ciliated cells. The cilia beat back and forth and circulate the cerebral spinal fluid throughout the central nervous system. The fourth and final neuroglial cell of the central nervous system is the oligodendrocyte, shown here in white. Oligodendrocytes have these arms that extend and wrap around the axons of nearby neurons, forming a myelin sheath. A myelin sheath insulates the axons in a manner similar to the way rubber insulates the wire component of an electrical cord. We will talk more about the function of myelin sheaths in a future screencast. So to summarize, the neuroglia or glial cells of the central nervous system include the astrocytes, microglial cells, ependymal cells, and the oligodendrocytes. The neuroglia of the peripheral nervous system include satellite cells and Schwann cells. Satellite cells insulate and protect the cell bodies of neurons in the peripheral nervous system. The second neuroglial cell of the peripheral nervous system are Schwann cells. Schwann cells form myelin sheaths and neurolemmas in the peripheral nervous system. If you look at this figure here, notice that the Schwann cells do not have processes that extend and wrap around the axons of neurons. Instead, the entire Schwann cell wraps around the axon and forms a component of the myelin sheath. Let's look at this in more detail. So Schwann cells wrap around an axon like a jelly roll. Notice that right up against the axon are very tight wrappings which only consist of the plasma membrane of the Schwann cells. And this is what forms the myelin sheath. Notice that the bulk of the cytoplasm of the Schwann cell is in the outer wrappings, and this is what forms the neurolemma. The function of the neurolemma is to protect the axon from physical damage or trauma. Remember that in the peripheral nervous system, we don't have the protection of bone like we do in the central nervous system. Let's now review the objectives of this screencast. Describe the structure and function of glial cells of the central nervous system, the astrocytes, microglial cells, ependymal cells, and oligodendrocytes. And describe the structure and function of glial cells of the peripheral nervous system, Schwann cells, as well as satellite cells. The next screencast discusses the topic of neurons.